Continuation of the previous video. This video is about cell-mediated immune response, NK and NKT cell function and mucosal immunity, part 2. And this particular study note is developed by EPG Panchala team, MHID. So, Government of India, I would like to express my sincere gratitude to the development team for developing this note. Thank you so much. Both specific and non-specific components of host protection from various infections lead to a state called immunity. The innate immunity is the non-specific component and considered as first line of host defense, while the adaptive immunity is a specific component with characteristic property of memory. The non-specific innate immunity consists of defensive barriers, which can be anatomical, physiological, or inflammatory in nature. Effective and first barrier to resist the entrance of most microorganisms are anatomical and physical. Among anatomical barriers, mucous membrane and skin prevents the penetration of exogenous microorganisms. Mucus is a viscous fluid secreted by the epithelial cells of membrane lining which entrap pathogens. Cilia are present in the mucous membrane, which helps in propelling the pathogens, competing with the natural microbes of the body for nutrients. And site of attachment. Mucous membrane is a major site for external microorganisms invasion. Mast cells, granulocytes, macrophages, and lymphocytes are residing in various regions within the mucosa membrane lining tissue. Intraepithelial lymphocytes are present in the epithelial mucosal layer. Mucosal immunity. Lymphoid tissue in lamina propria of lungs, intestinal wall, and other mucosal sites comprised of population of macrophages and lymphocytes. MALT or mucosal associated lymphoid tissues are less arranged and found at various sites of the host body. MALTs are associated in the intestinal lamina propria, upper airways, genital tracts, bronchi, and lungs mucous membrane linings, which provide protection against various viruses, bacteria, and parasitic infections. Majority of lymphocytes are B cell and T cell, along with activated Th cells and macrophages lying in lamina propria. All these functioning immune cells at the mucosal sites resist against infection and constitute the mucosal immunity of host immune system. B lymphocytes, IgA assisted mucosal immunity. An effective immunity of mucosa is dependent on the B lymphocytes assisted humoral response. IgA is a immunoglobulin molecule synthesized in the mucosal immunity. IgA antibody has to be transported to the respiratory tract mucosal surface, urinogenital or gastrointestinal mucosal surface through epithelial layer, which is dependent on secretory component or poly-Ig receptors present on the epithelial layer of these tissues. These secretory components are specific for the Fc region of IgA antibody molecule. When IgA molecule binds with the poly-Ig receptors present on the epithelial layer, vascular transport enables the export of this IgA-bound secretory component complex through the cell to the mucosal membrane. So, poly-Ig receptors gets partly cleaved by the proteolytic activity from the IgA molecule, and these immunoglobulin molecules, when secreted from the mucosal membrane, are able to neutralize antigens. So, lysozymes in mucosal immunity. Lysozymes were discovered in 1922, and the study suggested that the antibacterial action of lysozymes. Mucosal membrane of body secrete lysozymes, and these enzymes are found in the digestive system, saliva, mother's milk, tears, and reproductive organs. In the mucosal immunity, lysozymes are critically important proteins showing bactericidal activity. The mucous membrane of all these tissues, which are majorly exposed to microorganisms, consist of lysozymes protecting the host against infections. Effective and potential antimicrobial activity is contributed by lysozymes secreted from the mucous membrane. So, lysozymes attack the carbohydrate made protective layer on bacteria surface and thereby destroying the cell wall structural integrity and ultimately burst the bacterial cell wall. Then, gastrointestinal protecting role of lysozymes. Lysozymes improve the health of GI tract by competing with the pathogenic microbes and resisting their co-localization in the GI tract leading to the clearance of infectious bacteria. Lysozymes exhibit bactericidal activity causing autolysis and apoptosis of the invading bacteria. Lysozymes also stimulate other immune cells in the mucosa membrane to further provide protection against viral and bacterial infections. A potentially activated immune cells are IgA, AMPs, etc. Gastrointestinal protecting role of lysozymes. Lysozyme improves the health of GI tract by competing with the pathogenic microbes and resisting their co-localization in the GI tract, leading to the clearance of infectious bacteria. Lysozymes exhibit bactericidal activity causing autolysis and apoptosis of the invaded bacteria. Lysozymes also stimulate other immune cells in the mucosa membrane to further provide protection against viral and bacterial infections. The potentially activated immune cells are IgA, AMPs, etc. Then NK cells and mucosal immunity. Recent publication revealed the reclassification of innate immune cells into a large group based on the diversity of functional performed and development, which is termed as ILCs, innate lymphoid cells. So natural killer or null cells are first line of host defense system, which is non-specific but still acts potentially against virus infection and tumor growth or cancer. These are large granular lymphocytes sharing common origin from lymphoid progenitor, but they do not process antibody molecules bound to their membrane and TCRs like B cell and T cells, and do not undergo gene rearrangement or antigen receptors like other lymphocytes. They are broadly categorized into three major groups, group 2 and 3. The grouping systems of innate lymphoid cells are based on the regulation of transcription factors and functionalities of different cell type development. A conventional NK or null cell belonging to group 1 innate lymphoid cells. NK cells secrete a broad range of cytokines such as IFN, gamma, and cytolytic in nature. NK cells bind with the infected tumor cells to KAR molecules on human NK cell surface, and activated NK cells creates lysis or apoptosis of aberrant cells. NK cells play different but vital roles in maintaining the homeostasis of immune system, protection and modulation of immunological memory by activating other immune cells, thus provide long-term immunity. NK cells reside in many tissues of host including blood, bone marrow, thymus, liver and spleen. NK cells are also harbored at the mucosal sites such as intestine, lungs, colon and uterus, vagina, female, etc. We will be addressing only group 1 of innate lymphoid cell that are conventional NK cell in this chapter. NK cell development. Natural killer cell. Hematopoietic stem cell differential can lead to different developmental stages of human NK cell through a common lymphoid progenitor. There are 5 development stages at each stage. These cells acquire the expression of different cell surface markers. At stage 4, the cells are considered as immature NK cells as they express bright CD56 and high levels of cytokine, but they are still CD16 negative. At the developmental stage of 5, they become CD16 positive with dim CD56 with high catalytic and cytokine production, therefore considered as mature NK cells. And migration of NK cells to mucosal sites. Blood, bone marrow, peripheral non-lymphoid and secondary lymphoid organs are the residential sites of NK cells. Majority of human NK cells are immature with CD16 and bright CD56 expression in lymph nodes, while in lungs, guts and other non-lymphoid organs, morphologically different NK cells reside, differentiated on the basis of development and function. Once NK cells complete their developmental stages and exit bone marrow, they can either enter the blood or escape to secondary lymphoid structure. Modes of NK cell development suggest that a mixture of mature and immature NK cells emerge from the bone marrow. Immature cells acquire tissue-specific phenotype, and a mature cell under the influence of tissue microenvironment, growth factors, cytokines gets modified. The process of migration of NK cells from bone marrow to residence establishment in specific tissues is unclear. Recruitment of NK cells to mucosal tissues is the initial step towards its mechanistic studies. Then mucosal site homing by NK cells. NK cells employ different cytokine receptors such as CCR5, CCR2, CCR7, CXCRI, CR1 and CX3CR1. Additional factors include Cameron, SIP5 family members, etc. NK cells' role in pulmonary infection. Human NK cells play an essential role in protecting mucosa from infections. On pathogen-assisted stimulation, NK cells are recruited at the site of infection and produce TNF alpha and IFN gamma and display cytolytic activity. So NK cells can respond to diverse pathogen in different mucosal tissue sites by activating enzymes such as IL-15, LY49H, etc. and production of millennia of cytokines. NK cells are recruited to inflamed tissues on fungal, bacterial, parasitic or viral infection. Once NK cells are stimulated, they act as first line of host immune defense, thereby preventing the spread of infection. NK cells do not require any cellular interaction or specific signal for migration into lymphoid organs. NK cells, after completely or partly completing the developmental stages, giving rise to mixture of mature and immature NK cells in the bone marrow, through bloodstream migrate to lungs, where they exhibit CD16 plus DIM and CD56 expressions, indicating mature phenotype of conventional NK cells. NK cells' function and development are influenced by lungs microbial environment. Dendritic cells, also called antigen presenting cells of lungs located in the lamina propria, get stimulated after pathogen encounter and produce cytokines. These cells activated NK cells by binding human KIR molecules of NK cell surface and ultimately lyse the target cell by the secretion of cytokines IL-17, IFN gamma and granzyme. A major site of entrance for bacteria, fungus, virus and other infection agents is lung, and hence largest counts of group 1 innate lymphoid cells are present in lungs. Humans are more susceptible to lung infections on NK cells deficiency. Viral infection is very common through airways such as influenza virus, herpes, SARS and RSV. Seasonal and pandemic H1N1 influenza infection is controlled by the early response and cytolytic activity of NK cells to cytokine and IFN gamma production. Directly through natural cytotoxicity receptor binding to surface molecules of influenza virus, hemagglutinin, or indirectly through ADCC mediated or antibody dependent NK cells, cytolytic activity is a response to influenza virus infection. In addition to this, the response of NK cells to infection in lungs suggested the role of IL-12, IL-15, IFN alpha and IFN gamma cytokines. IL-15 along with IFN gamma and IL-12 activates NK cells in lung against endogenous bacterial infection via Staphylococcus aureus, which is a gram-positive bacteria. So cytolytic activity with the production of IFN gamma helps NK cells in early immunity and control of Aspergillus and Cryptococcus neoformans infection. Several bacterial infections such as Klebsiella pneumoniae, Staphylococcus aureus and Legionella can be controlled by the NK cell production of IFN gamma. IL-22 is an important cytokine produced by NK cells to control Klebsiella infection. So IL-12, gamma, IFN alpha beta are vital cytokines for the inhibition of Mycobacterium tuberculosis, growth and restriction of infiltration macrophages into lungs, which are produced by NK cells in response to MTB infection. Bone marrow NK cells, natural mature NK cells, blood, lung lamina propria, lung lumen. Illustration of role of NK cell in lung mucosa and female reproductive tract and NK cells. Immature NK cells interact with the dendritic cells of the gut and female reproductive tract located in the lamina propria, and immature cells homing are allowed to correct tissues by labeling the tissue addressed to the immature NK cells, such as homing to gut require alpha 4 beta 7 CCR9. Rest of the process of NK cell function is similar to that studied in lung mucosa. Enhanced response of NK cells and higher expression of IFN gamma levels may lead to loss of pregnancy. Gut mucosal infections and NK cells. Gut infections through Yersinia enterolytica, and Citrobacter reduction can be controlled by NK cell assisted IFN gamma production. Toxoplasma gondii leads to IL-6 dependent IL-17 production by the NK cells of gut mucosa. In the intraepithelial lymphocytes, NK cells mediated IL-22 and IL-17 produced through the interacting gut mucosal DC dendritic cell, which are CD103, can control early infection of HIV in humans. These NK cells residing in gut mucosa can potentially provide protection against infections which are orally required. NKT cells and mucosa immunity. Host interactions with its external environment with the help of mucosal surface, airway surface for gaseous exchange, gut and intestinal surface for essential nutrient uptake. The surface also acts as first line of defense, including the physiological and anatomical barriers against pathogens. Innate immune response includes the secretion of deficits from the epithelial cells of mucosal tissue. Mucosal associated antigen presenting cells like DC represent scavenger receptors and PAMP, pathogen associated molecular pattern and molecules to recognize pathogens. Besides, DCs also activate the T cell mediated adaptive immune response and humoral immune response through B cell mediated IgA response. A broad range of cytokines are produced from these innate or adaptive lymphocytes, resulting in enhanced immune response. So, innate lymphoid cells and natural killer cells are thus critical for the generation of mucosal immunity against pathogens. A minor subset of T cells represent natural killer NK cells, sharing some common surface markers with NK cells and T cells. Similar to T lymphocytes, NKT cells mature in primary lymphoid thymus tissue and mainly found in bone marrow, liver, and spleen. Among intraepithelial lymphocytes, invariant iNKT and variant natural killer T cells are abundantly found resident in the intestine, while mucosal NKT, mNKT mainly within the lamina propria of lungs. NKT cells represent T cell receptors on its surface, which binds to class 1 MHC-like molecule CD1D on antigen presenting cells. So, mucosal immunity in various mucosal membranes lining tissues are controlled by the activation of NKT cells via CD1D representing APC. Then development and differentiation of NKT. The sublineage of NKT cells include conserved transcription factors such as GATA3, TBET, and ROR gamma T. PLZF, promyelocytic leukemia. Zinc finger is a transcription factor which defines the developmental lineage of NKT cells. Various development stages leads to development and maturation of NKT cells in thymus tissue. Different developmental stages express different surface molecules and distinct transcription factors, leading to TH1, TH2, and TH17 differentiated polarized subsets of NKT population. Then next is activation of peripheral NKT cells, which we will be learning with the help of next video. Thank you for listening.